I came to India just 24 hours ago, and it's been already very delightful to be in this very nice environment. Now, my presentation will focus on the effect of dietary and cultural differences on the gut microbiota, and basically what I'm about to discuss with you is the mutualistic association derived from a long coevolution between human bodies, human cells, and microbes. And you can see it also as the interaction between the human genome on the one hand and the microbiome. So each of us is host to 10 trillion human cells facing 100 trillion dominant bacteria. And this is 23,000 human genes facing more than 500,000 bacterial genes in each of us. And overall, as we see it today, more than 5 million bacterial genes in the human microbiota. Functions that play those microbes are key at interfaces: food microbe interface, cell microbe interface, and microbe-microbe interface. And these interactions are essential to maintenance of health and well-being of humans. Food microbe interface relate to metabolism. This is the contribution of microbes to fiber breakdown, for example, which is the very tip of the microbe food chain in our guts. The production of fermentation metabolites that are key elements of the crosstalk between microbes and human cells, especially at the level of the gut epithelium, and also the bioconversion of xenobiotic and food constituents playing with the bioactivity of essential substances. At cell microbe level, this is essentially the maintenance of the mucosal barrier integrity in terms of quality and quantity delivery of mucus, but also tight junction integrity, trophicity of the epithelium, and all the parameters that relate to immune maturation and development, as well as newly recognized gut-brain crosstalk. And finally, microbe-microbe interaction that relate to essential colonization prevention, also known as barrier effect, very well seen for the prevention of pathogen proliferation, but which probably also plays a role in the ecology of the gut at large. My presentation will have three major parts. The first question is, what does the healthy adult intestinal microbiota look like? And I will give you the metagenomic perspective in that respect. Second, I will raise one question, which is, does diet and lifestyle impact human intestinal microbiota development, and how? And finally, I will stress the question, can diet be used to modulate the human intestinal microbiota? So for the first question, we entered, as was mentioned by Dr. Nair, the metagenomic era at the beginning of 2000, and the metagenome we are studying is made of the combined genomes of all dominant microbes within complex ecosystem. It was initially developed to study extreme environments, and it was over the years applied to the human gut microbiota. So I will take my illustration from the MetaHit European program, which was coordinated by INRA. We essentially need to have samples from which we extract DNA, and mention was made of standardization of procedures. I cannot emphasize this today, but this is a very essential element within the procedure. And then we use whole genome shotgun sequencing to generate short fragments of DNA that are then assembled into genes and annotated. And the result of this is the reference gene catalog and the possibility to develop very rapid gene counting technologies. The first reference gene catalog was produced and published in 2010. It was based on the characterization of 124 European individuals, and what we identified was a set of 3.3 million independent bacterial genes from these individuals. This mathematically was recognized to cover 85% of the abundant genes of the cohort and represented 150-fold the size of the human genome, which is shown here. And what was also important in this presentation was that the reference gene catalog, designed on the basis of characterization of European individuals, was very well applicable to characterize the data available for US individuals as well as Japanese individuals from the literature. The next question we raised was, is there a core, a set of genes that are shared among humans? From the characterized cohort, we indeed could identify a core metagenome. Each individual has on average 540,000, more than 500,000 bacterial genes out of the 3.3 million gene catalog. That in itself, considering we were characterizing more than 100 individuals, was telling us there were common genes between those individuals. And indeed, we found similarities. We found core metagenome genes such that approximately 50% of each individual's bacterial genes were shared by at least 50% of the cohort. So on that basis, we are all quite similar, but at the same time, we found individuality, we found rare genes that are shared by less than 20% of individuals of the cohort, and they represent 2/3 of the initial 3.3 million gene catalog. So we're not identical in that respect. And we do find genes and genomes that are subject specific for each person we analyze. Very recently, the paper of Loisnik and colleagues in Nature last month discussed of the metagenomic landscape of the system, and it just went further in supporting the concept that there is an intimate host microbiota mutualistic association that builds over the first years of life, and that remains fairly stable thereafter, even at the finest level, which is the gene sequence level. And then we raised a very simple question. If we have a core metagenome, then we should be able to identify the average human intestinal microbiota, which is a nice perspective when one wants to address the normal situation. And, well, we did not find anything like an average human intestinal microbiota. Instead, what we observed was 3 preferred combinations of microbial taxa among human intestinal metagenomes studied. And we call these the enterotypes. So individuals studied so far, and this has been observed repeatedly already since the 2011 publication. They separate in 3 enterotypes that are ecological arrangements that are dominated by the genera Prevotella for one of them, Bacteroides for the other, and Ruminococcus methanobrevibacter for the last enterotype. Not only do we see the dominant contributor to the enterotyping, but we also have an arrangement of bacterial genera that are either positively or negatively correlated with the main taxon. So people differ by species, they differ by enterotype, and they also differ by gut bacterial gene count. And I will really, over the rest of my talk, very often come back to this parameter, which in a way relates to bacterial diversity of the ecosystem. So the blue curve here is the distribution of humans as a function of bacterial gene count, and what you see is that there are individuals with low bacterial gene count and individuals with high bacterial gene count. In the overall population, here a cohort of 277 individuals, we find the spread quite large, from 200,000 to over 800,000. What's also important here is that, as you see in the brown curves, the distribution of the enterotypes is not homogeneous at all. The Bacteroides enterotype is overrepresented in individuals with low gene count microbiota. We have sequence information, so we can identify bacterial species that are overrepresented in one or the other of these phenotypes. So here what you have is a representation where each column is an individual. Each line is a set of 50 marker genes for each of a number of species. The top one are species for which we have genome sequence available, and you can see that this set of species is overrepresented among low gene count microbiota, and these are overrepresented among high gene count microbiota. So they are marker species for this parameter. Overall, we find 68 marker species that allow a stratification of the human population in low gene count versus high gene count. So let's switch to the first question we want to raise. Does diet and lifestyle impact human intestinal microbiota development? Obviously, we start with the very beginning. We are sterile in utero, and the intestine is colonized at the very first moment of birth. Intestinal colonization is affected by many parameters, including mode of delivery, vaginal versus cesarean section delivery, and maternal microbiome at the level of vagina, also intestine and skin. Hygiene of the neonatal environment and use of antibiotics will also play a major role, probably most importantly in the very first moments of life, but that will still last throughout the rest of life. And food and feeding mode, breast milk versus formula milk, but also weaning diet, will also have a strong influence. Now, I'm here citing a citation from Jean-François Bach, who coined the hygiene hypothesis, saying that early exposure to low microbial diversity would prevent or delay maturation of the mucosal immune system and thereby promote aberrant responses to allergens or autoantigens and associated lifestyle disorders, which were mentioned already, and for which we see this major increase over the second half of the previous century, at a time where we were regularly controlling better and better infectious diseases. So this was true for autoimmune diseases such as multiple sclerosis, type 1 diabetes, inflammatory bowel disease, metabolic disorders, obesity, type 2 diabetes, and allergy. Well, just going back to cesarean section, this statistics from year 2005, where I do not have India here, unfortunately, and I don't know where is the figure for India, but it states that the average for OECD countries was in 2005 approximately 20% of birth having recourse to cesarean section. Now, this will have an impact. Vaginal delivery leads to lactobacillus and prevotella as early colonizers, and vertical inheritance from the mother to the child at the strain level, whereas cesarean section exposes colonization to environmental microbes, and this is associated with higher susceptibility to certain pathogens and higher risk of atopic diseases, for example. Here what I emphasize is the fact that for some countries, the percentage of recourse to C-section is so high that it is my belief that this is beyond strict medical justification. And so my thinking as a microbial ecologist is, if we alter natural colonization and maturation, then we will have missing components, and nutrition will not be able to restore any element of distorted situation, especially when we address the intestinal microbiota. Maybe this is related, I cannot be certain, but in this publication from May 2012, the group of Jeffrey Gordon compared the microbiota in terms of species richness between US citizens and subjects from South America, Amerindians in Venezuela, and Africa, Malawi. And what they showed is, although from birth to 3 years of age there was not much difference, after the age of 3, the American subjects are all showing less species richness, i.e. less microbial diversity in their gut microbiota compared to any of the other citizens from South America and Africa. Now, we have been in developed countries impacting probably over several generations microbiota and microbiota transfer from mother to child via nutritional transition, via repeated antibiotherapy, and probably other parameters related to hygiene at birth. My question would be, have we been altering the human biology in altering the human gut microbiota? I will leave it as a question. Dietary habits has been shown, and this is recent work from De Filippo and colleagues in Italy, that they impact or they shape the microbiota of infants. The difference here is between microbiota in kids from Burkina Faso in Africa and Italy in Europe. And you can see the difference is striking. There are differences at the level of phyla, Bacteroides and Firmicutes, Bacteroidetes and Firmicutes, but even within the Bacteroidetes, the main phyla or the main genera is Prevotella for the Burkina Faso children, whereas it's Bacteroides for the European infants in Italy. So main impact of diet. After the publication describing the enterotypes, we knew that human population would stratify in 3 enterotypes, but there was and there still is no genetic basis for it, and there is no ecological meaning that has been yet ascertained to explain that. So Wu and colleagues published in Science 2011 a characterization of the microbiota of different human individuals in the US, where they would see 2 enterotypes very nicely, the Bacteroides and the Prevotella, in a US cohort. And they showed that long-term dietary patterns do shape the enterotypes, such that the Bacteroides enterotype is highly represented in individuals with high animal protein and fat diet, the Western type diet, I would say, whereas Prevotella enterotype is highly represented among high carbohydrate, high fiber diets. At the same time, in this study, they reported that there was no modification by short-term dietary intervention, a 10 days intervention. So if we summarize, take home messages from this part, then among several factors, diet will indeed contribute to structure the microbiota, especially in the early moments of life, and there is an impact of long-term dietary habits on the separation of humans into enterotypes. Dietary changes may play a role over several generations, probably more than we have thought of, and once established, the adult microbiota composition, at the level of composition, if I do insist, will be fairly resistant to moderate dietary changes. And I will finish with this question: can diet be used to modulate the human intestinal microbiota? And here we do need a concept, a functional target and readouts, and the example I will take is response to diet in obese individuals. And I will use for my presentation work done in 2 European studies. But first, the context is that we know there is differences in the microbiota in obesity, and from the work of Jeffrey Gordon's group, especially that piece of work showing that there is transplantation of the microbiota that allows to transplant the propensity to fat increase upon diet-induced obesity conditions. So mice, germ-free mice that receive the microbiota of conventional OB mice will have a tendency to take up fat mass more rapidly than the same germ-free mice receiving the microbiota from conventional mice. And there are differences in the structure of the microbiota between the 2 types. In Europe, we had two studies, the European MetaHit study, which involved close to 200 individuals, lean and obese, and was coordinated by Oluf Pedersen. It was based on a Danish cohort built in 1999, and so people were followed over time for close to 10 years before we sampled their microbiota. In French individuals, we had 50 obese and overweight that had a calorie-restricted diet for 6 weeks and a following maintenance diet for 6 weeks under supervision by Karine Clément at Pitié-Salpêtrière Hospital. Now, in both studies, what was quite striking is that if we study the distribution of individuals as a function of gene count, then we have low gene count individual, we have high gene count individual, and the distribution is actually bimodal. So we have, in a way, 2 separate populations. And it's also true in France, where the numbers are not so high, but the observation is the same, and the proportion of low and high gene count individual is the same as well. What was striking in these pieces of work is that low gene count individuals, low bacterial richness microbiota, have less healthy metabolic and inflammatory traits. And I will illustrate this in the following slides. So this is a bit complicated slide, but we will focus on the yellow highlighted elements. If we compare low gene count and high gene count individuals, then what we see is that insulin is significantly higher in low gene count individuals. Triglycerides, blood triglycerides are higher, and high sensitivity CRP, a marker of low-grade inflammation, is higher in low GC individuals. So overall, we have increased adiposity, we have increased insulin resistance, dyslipidemia, inflammation, all leading to predisposition to comorbidities in obesity, such as type 2 diabetes, cardiovascular disease, and possibly cancer. We also saw that in low gene richness microbiota, there is more frequent obese individuals, and it's associated with higher weight gain over the past 9 years for the Danish cohort subjects. This is illustrated here. If we take the overall population and separate the low gene count and the high gene count, then the percentage of obese is close to 80% in the low gene count individuals. It's lower in the high gene count, and this is true for male and female as well, where it's significant in both cases. And if we look at BMI change over time, then we see that in obese individual, low gene count comes with a higher increase in BMI over the past 9 years compared to high gene count individuals. So those individuals with low gene count will have a tendency to gain more weight or to gain weight more rapidly over time. In the French study, we had this intervention, 6-week intervention with very low calorie diet, 1,200 kilocalories for female per day, 1,500 for male. And this was a high protein, low glycemic index carbohydrate with high and diverse fiber content. And then stabilization. And what we could see is that the dietary intervention will altogether improve gene richness. This is shown here, where over time, if we compare low gene count and high gene count, what we see is that we can rise the gene count in low gene count individuals by close to 100,000 genes. So this is very significant here. But at the same time, although we do correct the phenotype partially, we do not correct phenotype as well in the low gene count and high gene count individuals, such that the insulin resistance, the triglyceride blood levels, and the high sensitivity CRP are not ameliorated as well in low gene count individual. Overall, the conclusion is that dietary intervention appears less efficient in patients with low gene richness, especially for inflammation parameters. But I will stop 30 seconds here. What I'm saying is that if we take the picture of the microbiota of an individual before we do the intervention, at time 0 of the study, we can predict whether that person will respond more or less to a very drastic calorie restriction. So this is predictive here. Now in this study, we worked with colleagues from Danone on analyzing the dietary habits of the patients, and using dietary patterns, we determined 3 different patterns based on food frequency records for 26 food categories. We found a cluster 1, cluster 2, cluster 3, 1 being characterized by consumption of potato, including chips and sweetened drinks, for example, whereas cluster 3 was characterized by consumption of high fruits and vegetables in their diet. And there was a link in this study between the category, the cluster of dietary pattern, and gene richness. It was only a trend with a p-value of 0.01 for the small population that we were analyzing. But we think that healthier dietary habits being associated with higher gene diversity could explain, at least in part, the more favorable phenotype of the subjects with high gene count microbiota. Hence a potential major role of nutrition on the phenotype and the development. So if we look for take-home messages of this part, microbiome gene richness appears as a key stratifier for the response to dietary intervention in obese subjects. Microbiome gene richness is a relevant target for dietary modulation, and this was not so clear until today. And dietary modulation of microbiome gene richness is possible, as far as what we have seen, on the basis of low glycemic index, high fiber diet at least. And to conclude, just one more question. Is there a place for functional foods? We've learned from work by Patrice Cani and colleagues that prebiotics are successfully used in modulation of low-grade inflammation in obesity and type 2 diabetes. The concept is that diet modulates the microbiota. This biosis of the microbiota changes barrier functions, especially with the modulation of endocannabinoid gene expression and tight junction, and it induces endotoxemia, inflammation, and thereafter metabolic syndrome. We also know that probiotics are successfully used in modulation of allergy, immune homeostasis, and gut health. At the European level, regulation is being centralized, much like you have many states and you do efforts to centralize the regulation of health claim in India. And at present, it's in major expectations for the regulations of health claims towards a general consensus, and the rule will help the EFSA, the European regulatory body, to basically help the population decide on what are the valid allegations in that respect. There are novel concepts that should be open perspectives for new targets. I mentioned diversity quite a lot, and I have one more slide to illustrate what we can expect on the stability itself with probiotics. Stratification of responders and non-responders may also be an important point. And microbiota modulation should really only be considered when we have balanced nutritional conditions, I would say. So the last slide here, we studied probiotic administration and microbiota stabilization in the context of ulcerative colitis, an inflammatory bowel condition. In this context, people received on a random basis either a placebo or probiotic for a period of 3 months, and we looked at stability of the microbiota based on metagenomic profiles. What we did see is that if we take the overall population, and this is work that was supervised by Francisco Guarner's team in Barcelona in conjunction with colleagues of Danone Research, we see no significant impact of the probiotic versus placebo on stabilization of the microbiota. But if we only consider the high gene count population or microbiota population, then we have a highly significant impact of the probiotic administration on stabilization of the microbiota. So here again, we're into conditions where we can predict the response to a probiotic administration in patients in terms of stabilization of the microbiota by probiotic administration in this case. So diversity-based stratification of responders, non-responders is quite instrumental in this condition. So I would like to acknowledge the contribution of the many colleagues of INRA who took part in the work that I presented, the partnerships of MetaHit and Dushko Erlich, who is my colleague of INRA who coordinated MetaHit Consortium, as well as Karen Clément, who coordinated the micro-obese study, and the many clinical doctors with whom we've been working in the Paris area and in Europe. And I thank you. Merci for your attention.
Thank you very much, Dr. Dore. That was a great start.
Thank you.
The talk is open to questions. Yes, sir.
May I first pay compliment to you, Mr. Dore,
Thank you.
for tackling a very complicated subject in this small time, short time that you had at your disposal. Well, I'll pose two issues. First is that you very rightly hinted that there is the role of the bacterial genes and microbiota in maturation of the immune system.
Yeah.
Of course, the diversity we know is to a great extent driven by that phenomena. Also, a very important facet is that the oral route is the one which leads to tolerance or lack of immunological responses. I mean, that is again a very great feature. And that is what avoids the autoimmune phenomena. Otherwise, we will be all allergic to chicken, fish, and God knows what.
Yeah.
So therefore, this is a very important attribute of the system. Do we know which microbes are connected with these two phenomena? This is the issue that I would pose. Second, a little comment is that for the metabolic syndrome, please don't forget there is also a hormonal angle. It is not just the number of genes, the high or lower, but also the hormonal
Yeah.
circuitry,
Yeah.
which has a very important role
Yeah.
in the metabolic syndrome.
Okay. Thank you very much for these questions. And I think both relate to potential crosstalk between microbes and human cells. So you will hear more from the presentation of Jean-Michel Antoine on crosstalk. But what I would like to say is that we know very well the interaction between pathogens and human cells and how that relates to immune system. But we know still rather little about the interaction between human commensal microbes, dominant commensal bacteria, and human cells of the epithelium as well as immune cells. Yeah, so the way we address this today is using the metagenomic approach, and basically we extract the genomic information from the human colonic microbiota, and we clone this information into E. coli carriers, for example, and then we can put in contact those E. coli clones, metagenomic libraries, together with human cells that have been engineered to emit light or color when there is interaction that relates to immune modulation. And when we explore this, as well as when we explore culture collections of commensal strains or probiotics, what we see, for example, addressing NF-kappa B modulation, which is a basic evident target, then we see that we have microbes that activate or stimulate NF-kappa B response, and we have microbes that down-regulate NF-kappa B response. So we can actually identify genomic fragments from the metagenome or strains from dominant commensals or probiotics that are active in talking to human cells. It's a defined system. It's an artificial defined system, I should say, but still that allows to identify who's talking to human cells in relation to immune modulation. And we're quite optimistic that that will lead us to understand better which are the major actors in terms of commensal bacteria and also among collections of probiotics, for example, and what are the molecules of the crosstalk. Those we know well for pathogens, but we don't know so well for commensal bacteria. So this is work in progress, I would say. At present, in my group, we are addressing that very closely, and we have out of probably 20,000 or more metagenomic clones with fragments of 40 to 50 genes of gut bacteria, some many probably uncultured. We have identified 20 to 30 metagenomic clones that do talk to human cells. The reason why I started saying that both of your questions possibly relate to bacteria host crosstalk is that also the hormonal important parameter for food uptake regulation, for example, that you mentioned, that might be under, at least in part, under regulation by bacteria cell crosstalk at the level of the epithelium, where bacteria are sending signals to enteroendocrine cells, for example, and that will govern PYY and GLP-1 production, regulation of expression, and hence possibly neurological responses. So I think that your topic is very important. It's emerging, and we can hope a lot from that. Yeah.
Yeah. I'm Dr. Varsha. I'm a clinical nutritionist, and I deal with the pediatric population as such. And my observation is, and I stand vindicated with what you have presented today, I have mothers who deliver in the US or Europe and come back with children with problems, back into India. And what I have been recommending to them till now is that the mother continues to give the child food that they are more familiar with back home from India, rather than accepting food that is available to them in the US or UK. And I find that it gives better outcome and results to this scenario where we have problems. So would it be right for me to conclude that, you know, when it comes to the vaginal or versus the LSCS deliveries, the C-section deliveries, and then when it comes to the maternal diet and what needs to be given to the infant, we continue to give them the same environment?
Yeah. So it's an important topic, really, and I'm not a pediatrician, so we're in a specialist area, but still my feeling as a gut microbial ecologist is that any change will have an important impact possibly on the microbiota, and as I try to emphasize dietary changes, even the mother diet when she's breastfeeding, for example, will possibly have an impact on the microbiota. So basically, the less you change the habit, the less you stress the microbiota. And so especially if there is a country change, which is already probably stressful for the microbiota, then accompanying that with the least possible changes in the diet probably would help keeping the homeostatic condition that is being built. So I think that your recommendations make a lot of sense for me, actually.
Thank you.
Okay, just to keep locational balance, one from General Raghunath. We'll take two more questions after this.
Yeah. May I add my compliments to a very nice and fascinating lecture. Right? Dr. Dora, it was very interesting. I was just wondering whether these enterotypes have any balance with the racial structure of humanity.
Whether they are balanced with?
The races, human races.
So from what we know today, I must confess that it's more driven from the characterization of the Northern Hemisphere sampling cohort, I would say, and there's data from US, Japan, and Europe that are dominant in the data we have at hand. So we may have not a complete picture because there is very little data coming from Africa or other Asian countries than Japan for the moment.
The racial diversity that you saw apparently is not indigenous, and therefore it will not be relevant. Would that be right?
Yeah, I would say yes, yes.
Professor Agarwal.
My question is focused to the last part of your talk.
Yeah.
The study on giving probiotics to people with ulcerative colitis.
Yes.
You said you used the term stabilization of microflora.
Mm-hmm.
What exactly does that term microflora stabilization mean?
So we looked at the picture of the dominant gut microbiota. And when you do this over time in UC patients, what you observe is a very unstable condition. So just basically comparing the overall, in this case, metagenomic picture, you see that there are changes in genes and that basically correspond to changes in dominant microbial species. And so we were looking at the percentage of similarity over time with time zero, basically.
So, is there a difference in ulcerative colitis versus healthy people in this stability?
Yes, there is. There is, yeah. I had in my slide comparison with the healthy controls, and the microbiota in healthy individuals is very, very stable over time, actually, unless we have antibiotic treatment or major stress in terms of fiber intake, for example. Otherwise, it's remarkably stable over time in healthy.
We'll take one more question after this, Dr. Bhatnagar.
Go ahead.
European, you selected only 124. I mean, how did you select this sample of defining healthy individuals?
Yeah, why?
That is one. And secondly, does the metagenome of an individual remain static over a period of time, or does it change?
Okay, so for the first part, why did we select these 124 individuals for the enterotyping? Actually, we at some point we are including subjects for the MetaHit study from Denmark and Spain, essentially, and we analyzed the data as it was coming, and when we had a clear view of the enterotype, then we stopped the analysis, but we have been continuing all the time ever since. And today we have metagenomic information from close to 1,000 individuals, and we'll still see very clearly the enterotypes. It is a debate within the scientific community as to the...
My question is, adding these more individuals, is the diversity increasing in the metagenome, or
Yeah.
is it becoming a pattern?
Yeah, so do we see an increase in diversity as we analyze more and more individuals? So what we see, unfortunately, I should say, this is still within European or Northern Hemisphere populations, Western style, but we do see an increase. So the reference catalog, which was 3.3 million when it was initially published in 2010, today is probably close to 8 million, but the more genes we see are actually essentially rare genes. So we do find always the core metagenome, and we increase our understanding of the rare genes as we analyze more people. But we still keep this same vision of a core metagenome and 3 enterotypes. And
Dr. Sanjini Bhatnagar and then Dr. Prajapati. This is an area where more questions than answers can be generated. So.
No, I just have a quick question. You know, this probiotic science is really wonderful, but we've been at the other end as clinicians and pediatricians, we've struggled to find cause-effect relationships. And I just had a question about your data initially. These were all cross-sectional data analysis with the diet and the diversity. I mean, when you're correlating or finding associations with diets, these were at one-time analysis, or it was because we don't have much data about long-term follow-up of maternal diet on infant microbiome or infant follow-up and their flora changes.
What I have shown can be viewed as essentially a one-time point in terms of nutritional information analysis. Although it's based on questionnaires to evaluate feeding habits more than just a one-time point feeding regimen.
Yeah, I was just thinking that, you know, that getting more data in a time series way would help us to understand these questions a little better for interventions eventually.
Yeah.
One would imagine that to happen in the near future because of the technological advances. At this point, it's still very expensive. Deep sequencing is still very expensive. Professor Prajapati, and we'll end up the session.
Your last slide was very interesting because probably you are drawing a conclusion that probiotic intervention doesn't affect much the number of microflora rather than the number of genes are more important, because all probably microbiomes are targeting high number of viable cells to reach to colon. So is it not that number which is very important, or the number of genes related to some effects? I could not understand much your last slide, that your conclusions in that.
Yeah, so if I understand well, you're asking whether numbers of genes more than species diversity is important. And I must say that as we see it, when I talk of number of genes, it's number of independent different genes. So they actually are directly connected to number of genomes that are dominant, hence number of species. So both are connected. It's just because we analyze the metagenomic profile that we can actually talk of number of genes. But if the resolution of the technology you may be using is only telling you of the species, you still see the difference in terms of species richness, hence diversity. So you may miss some connections if you have a less high resolution approach, basically. But it will be in most cases directly connected.
Thank you.